Hey, thanks for joining this session on biology. This is going to be about the beginning of chapter 13. We're going over the first two sections. And this chapter is about RNA and protein synthesis. So our last chapter was all about DNA, and this is about its partner, RNA. So first, let's kind of go over what it is and how it's similar to DNA. Both DNA and RNA are made of long chains of nucleic acids. They have nucleotides, which have a similar structure. You've got a phosphate and a sugar, and that kind of makes the backbone of the DNA and the RNA. And then a nitrogenous base that's hooked to the one side of it. And the nitrogenous base is where the code is in the DNA and the RNA. So the nitrogenous base by itself isn't much of anything. There's four types. You've got A, C, G, and T in DNA, but the order of the nitrogenous bases is what makes the code. And RNA uses the same code and just puts those bases in order, and that's how it's able to make things happen. So they're both built of these nucleotides. Okay, a nucleotide is just this whole unit, the phosphate and the sugar and the nitrogenous base, and they're all chained together, and they make the code out of the order of nitrogenous bases. Now, the way that they are different is the sugar part of the nucleotide is slightly different between DNA and RNA. The DNA is a deoxyribose sugar, and the RNA is a ribose sugar. So that's how they get their names. That's where the D and the R come from, DNA and RNA. Oops. DNA is double-stranded, and RNA is single-stranded. So it doesn't have two matching strands that are complementary that twist around each other like DNA. You just got one strand. And then one of the nucleic acids in the code is different. So in DNA, we have C, G, A, and T for cytosine, guanine, adenine, and thymine. But in RNA, that T is changed to U for uracil. So RNA has cytosine, guanine, adenine, and uracil. So it's the same code. It still matches up the same way where G and C bond together, and in DNA, A and T bond together, but with RNA, it's just A and U. It's just a replacement. So what is the point of having these two things that are so similar? We'll find out as soon as we do this review question. So which two of these statements are true? So we have DNA and we have RNA. A, DNA and RNA have different sugars. B, DNA and RNA both have adenosine, cytosine, guanine, and thymine. Or C, DNA has two strands and RNA only has one strand. So I'll give you a few seconds if you're watching the video and just pause it and think about the answer. So which is true? The first one is true. DNA and RNA have different sugars. That's how they get different names for the molecules. The next one is not true. DNA and RNA both have adenosine, cytosine, and guanine, but thymine is different. DNA has thymine, and in RNA, it's replaced with uracil. So instead of T, we're going to see U in the code. And then the third one is also true. DNA has two strands stuck together. RNA only has one strand. So we have these two molecules that are really similar. They both carry the same code. So why do we have two of them? What's the point of having these two different things? Well, they serve different functions. The DNA stays in the nucleus where it's safe. And there's only one copy of DNA, and it's all in one place. And that's important because that way when the cell divides, the nucleus is copied, and then each new daughter cell gets a copy of the DNA. And that way each cell isn't getting like multiple pieces of DNA or getting like some chunks but not the whole thing. It just stays right there in the nucleus where it's safe. It's kind of the master plan that you don't want to get screwed up, so it stays there and then just gets completely copied over so that each cell has one. With RNA, you get pieces of RNA that actually just move all around the cell to get things done. The RNA molecules are made from copies of the DNA, and there's many pieces of RNA in the cell at any time. So that's important difference in their function. Now, in DNA, the two strands of DNA are complementary. The code on one side fits together with the code on the other side using base pairing rules. So remember, with base pairing rules, um, A bonds to T and T bonds to A, and C and G bond together. So that's how we make sure that both sides are complementary. They follow those rules. Now, in RNA synthesis, the RNA that is created is also complementary to one side of the DNA, following that same base pairing rule, except that T is replaced with U. So G and C still bond together, and now A and U bond together. In this way, the RNA still carries this pattern that's related to the code in DNA. This process of making a strand of RNA is called transcription or transcribing. Oh, and there's our rules again. G and C bond together, A and U bond together. And because the DNA or the RNA is complementary to the DNA, it still carries that code. Now, when the RNA is being created, it's called RNA synthesis, and then, like I just said, it's made up of a process called transcription. So what happens is you've got your DNA. It's only one side of it. It's got these kind of naked nitrogenous bases hanging out here that don't have anything paired with them. So nucleotides of RNA come in and follow these bonding rules. So if the DNA has an A, then U has to bond to it. If the DNA has a C, then G has to bond to it. So they just come in, follow the base pairing rules and bond, and now we have a whole strand of RNA. So again, this process is called transcription. So it's pretty similar to DNA synthesis in that it's just bringing in matching nucleotides and making a chain out of them. Now, RNA kind of has a lot of jobs. There are three different types of RNA. Ribosomes are organelles that are made up of proteins and ribosomal RNA. So they're always shown having two units stuck together, and each of those units is both protein and RNA. Then we have messenger RNA, and that comes in carrying the code from the DNA to the ribosomes. It's going to kind of tell the ribosome what to do. And then you have transfer RNA, which brings in amino acids to the ribosomes. So we'll talk about why that's important in a minute. First, another review question. Which strand of RNA would be complementary to the DNA strand with the template with the code GTGACC? So if our DNA is GTGACC, what will the complementary RNA look like? Again, I'll stop for a few seconds. If you're watching the video, pause it and think about these answers. Which one's correct? So our first option, GUGACC, that doesn't work because it's not complementary. Remember, each one doesn't bond to itself. You don't have a G bonding to a G. G always bonds to C. So A can't be right because it doesn't use those base pairing rules. Okay, we don't have G bonding to C and A bonding to U. How about B? We've got G bonds to C, T bonds to A, G bonds to C. But then here's our problem. We have A bonding to T, and it should be U. So in RNA, we're never going to see the letter T. It's always U. So this makes this our answer. And I put it up here too. So if our DNA template up here is GTGACC, it bonds like this. G bonds to C, T bonds to A, G bonds to C, A, remember, bonds to U because it replaces T in RNA, and C bonds to G and G again. So you may be asked to do this exact same thing on a test where you're going to have a template like this of DNA and it says, well, what RNA do we get from that? Now, why does all of this matter? What is this RNA code for? Well, the code on RNA is used to figure out how to put amino acids into a certain order. So the DNA code is transcribed to RNA, and the code on the RNA codes for the order of the amino acids. And this whole thing is called translation. So making the RNA was transcription. Now making the chain of amino acids is called translation. And instead of chain of amino acids, the word for that is polypeptide, but that's what it means. You take a bunch of amino acids, you hook them together, now you have a polypeptide. And the point of them is that polypeptides make proteins. So you get a few chains of amino acids, they come together, they fold up really weird, and they make proteins. Now, the RNA is read in sets of 3. Each set is called a codon, and different codons code for different amino acids. Now we have charts that tell you how to go from RNA to amino acid. If the chart is round, like your one in the textbook on page 311, you need to start from the center and work outward to the name of the amino acid. So if it's CAC, you start in the center at the C, and then you go from that area, you keep going out to the A, and then the C, and then you're at histidine. So we're going to do a quick thing where we're going to an activity that's going to show how to do this. I just need to bring that up. Okay, so we're starting with this sequence of RNA, the one that's GAC. So how in the world do we turn GAC into an amino acid? Well, you have to look at your chart. So we start at the middle, so our first letter is G, so we're going to start with the G. Okay, so our next letter is A. So we just look at the ones that are touching this G over here and look for A. You don't want to jump all the way across and say, let's use the A from the other side. That doesn't make sense because we already picked G. So we start with our G. It's touching a U, C, A, and G. We want to go to the A that it's touching. And then our last letter is C. So from this A, it's touching a U and a C and an A and a G. We say, okay, we want the C that is touching. And that takes us to the name of the amino acid, which in this case is aspartic acid. Okay, so what you'll see is a chain of RNA code where you just have lots and lots of letters. You break them up into 3s. So we're starting with AUG. So you go from the A, the U that's touching it, and then the G that's touching that U. And that, I know it's very small letters, but it takes us to methionine. So that would be our first one. And then you just keep going, reading 3 of them at a time and saying which amino acid is coming out of that codon. And those get chained together, and that's what's going to end up making the polypeptide. We'll do a practice question in a minute, but there's also this idea you need to know that you've got really long chains of RNA. It's got to know when to start and stop. So there's certain codons that do that. So if you see an AUG, that's the start codon. That's always the signal that this is where we're going to start building our chain of amino acids. And then there's 3 different stop codons. You could have UGA, UAG, or UAA. That's where it gets cut off and stops adding amino acids. Oh, okay. And this is how we would find the stop one. So it's UAG or UGA. Those both lead to one that just says stop. So it doesn't actually code for amino acid there. It just codes for, okay, we're done now. You can let go. So let's do a quick practice question. We have this RNA sequence of UGG-GUA. So what two amino acids does that make? Well, first of all, what do you mean two amino acids? We have 6 letters there. Well, remember, it reads in codons. It reads in groups of 3. So UGG is going to be one amino acid, and then GUA will be a second one. All right, so UGG. We're starting at the U in the middle. So we can't jump across the wheel. We've got to use the ones that are touching this U. So we've got U, and then G, and then that's touching a U, C, A, and G. We go to the G, and that takes us to tryptophan. So that's our first one. And then our second one, GUA, we take the G and go to the U, and then go to the A. And that tells us it's valine. So that's our answer. We have tryptophan and then valine. Now, in real life, there's going to be hundreds or thousands of these in a row, but, we're just going to work with 2. Now, translation is where all 3 types of RNA come together. So remember, we said before, there's 3 types. Our RNA is part of the ribosome, so that's bunched up in here with some proteins. You could think of the ribosome as being the machine that makes polypeptides. Then you have the mRNA that comes to the ribosome carrying the code from the DNA. And then you have tRNA that brings in amino acids. And they have to match up with each codon. So we just saw that there's 20-some different amino acids. How does a tRNA know which one to bring? Which one's going to match up correctly? Well, the tRNA has 3 nitrogenous bases that are complementary to the codons on mRNA. And these are called anticodons. And the anticodon determines which amino acids the tRNA carries. So it allows a tRNA to bring an amino acid and bind onto a codon on the mRNA, one that it is perfectly complementary to. And so it brings the correct amino acid in place, and the ribosome keeps the amino acid, and it's going to bring, for our next codon here, we have this AGU codon. So the tRNA is going to bring in another amino acid. Because the tRNA has that anticodon, so it's looking for that specific AGU code, it matches it up, and the ribosome hooks the amino acids together, and now we're building a chain of amino acids. So it just does this hundreds or thousands of times and just keeps bringing in amino acids. So we'll go through all this to discover that the main purpose of DNA is just for providing a template for hooking amino acids together, making proteins. You have your DNA, which makes RNA, and RNA leads to chains of amino acids called polypeptides, and polypeptides fold up and make proteins. And that's it. That is what DNA is for. It codes to make proteins. And proteins run the whole body. So we focus a lot in biology on enzymes. They are proteins that control chemical reactions. And those control everything from the growth of tissues in the body to what color those tissues are, to the ability of a cell to break down and use nutrients at any given time. It's all controlled by enzymes. So your DNA is the blueprint for making these proteins so that they could do all of the jobs in your body. So that brings us to the end of this section. We're going to have another video where we finish up the chapter with section 3 and 4. But if you have any questions on this, don't hesitate to contact an instructor.